monoclonal antibodies had a major impact in the natural history of the B-cell malignancies, being rituximab the first monoclonal antibody to target the CD20 molecule. Rituximab was also associated with chemotherapy and became the standard of care in non-Hodgkin's lymphoma and in CLL, as you can see from these very important studies that were done. Uh, but we know that a proportion of patients, they do not respond to the association of chemotherapy plus rituximab, or they relapse to this treatment. And in these patients, we need, sorry. In these patients, we need new options to obtain a significant prolonged first remission in follicular lymphoma and for relapsed disease. Also, in diffuse large B-cell lymphoma, not all patients are cured. Therefore, new treatments for relapsed patients and improved first-line treatment for high-risk patients are needed. And particularly in CLL, we have a very special situation because many patients are elderly and have comorbidities and are not fit to receive the best chemoimmunotherapy that we have, that is FCR. For these patients, we also do not have still a good standard for treatment. And for fit and non-fit patients con deletion or mutation TP53, we also need new alternatives of treatment. Other CD20 monoclonal antibodies were introduced in the to treat this kind of patients. And of those, ufatumumab and obinutuzumab are already CLL approved in United States and the European Union and in several other countries. Studies on non-Hodgkin's lymphoma are ongoing. There are new monoclonal antibodies, veltuzumab, ocaratuzumab, and obinutuzumab, that are in early stages of phase 2 or early stages phase 3 studies. Sorry. The complement one study showed ofatumumab plus chlorambucil versus chlorambucil, this comparison. And in this study, in first-line therapy, unfit patients, CLL patients, we could have see a higher overall response rates and also complete response rates were compared to chlorambucil alone. The progression-free survival also was higher, significantly higher than when chlorambucil alone was given. There was no significant overall survival in advantage for ofatumumab plus chlorambucil versus chlorambucil. But still, the data may be immature. The endpoint for overall survival was not reached in either treatment group. Is well known the CLL study did by the German CLL study group. In this study, the study was divided in two parts. Stage 1 study compared obinutuzumab plus chlorambucil or rituximab plus chlorambucil with chlorambucil alone. And in the second part of the study, rituximab plus chlorambucil was compared to rituximab plus chlorambucil. In the first part of the study, there was a significant improvement of the overall response rate with both monoclonal antibodies, obinutuzumab and rituximab plus chlorambucil compared to chlorambucil alone. Also, when progression-free survival and overall survival was analyzed in this part 1 study, we could see that obinutuzumab plus chlorambucil and rituximab plus chlorambucil provided progression-free survival advantages over chlorambucil alone. And also, the combination of the monoclonal antibody plus chlorambucil showed overall survival in both groups compared to chlorambucil alone. Part 2 of the study also showed higher overall response rates, complete response rates, as well as minimal residual disease clearance, better than when we used obinutuzumab plus chlorambucil compared to rituximab plus chlorambucil. Obinutuzumab plus chlorambucil induced deeper and longer remissions than rituximab plus chlorambucil. Regarding overall survival, there is no significant difference between the two groups, but data are requiring additional follow-up. New molecules are available for treatment, and they were already extensively presented in this meeting. Ibrutinib, idelalisib, BCL2 inhibitor. Also, there is a new monoclonal antibody, polatuzumab vedotin, that is a monoclonal anti-CD20, anti-CD79B monoclonal antibody associated with a drug chemotherapy. I will not going to extend my presentation here because this was already presented. Ibrutinib was compared to ofatumumab and really significantly improved the progression-free survival and overall survival were obtained in this study. Idelalisib plus rituximab was also compared to rituximab alone, and the progression-free survival really was much better, including the overall survival when these two groups were compared. Antibody drug conjugates are being testing based on the results that we had with brentuximab vedotin in Hodgkin's lymphoma and the CD30 positive non-Hodgkin's lymphoma. Polatuzumab vedotin is a monoclonal antibody conjugated to the microtubule disrupting agent monomethyl auristatin E, and is good for the treatment of indolent and aggressive non-Hodgkin's lymphoma. But best results are obtained when this monoclonal antibody is associated with rituximab. Unfortunately, there were no objective responses in CLL. So we have studies 1, 2 that are ongoing. In conclusion, we have very new drugs to treat CLL, mainly, and also non-Hodgkin's lymphoma. But we need new agents, particularly ibrutinib, to see if they are more effective than conventional therapies in patients with deletion 17p and mutation TP53. But in this kind of patients, the follow-up is still short. We need to have more time to see really what is going to happen in these patients. Also, highly effective, less toxic therapy for typical elderly CLL patients with comorbidities are needed, and better therapy for high-risk CLL and patients with relapse refractory disease. Also, what is very important is to have a systematic evaluation of novel treatments compared to previous standard in clinical trials, and an extended time required to evaluate properly progression-free survival and overall survival. Open question. They are already in discussion until now in the meeting, and the problem will be more is if we have to eliminate chemotherapy or now replace it with antibody drug conjugates, if the if the combination therapies with new CD20 antibodies plus target therapies like anti-BCL2, BTK inhibitor, PI3K inhibitor, and also anti-PD-L1 offer high efficacy in the future. The role of MRD in CLL, if it's possible to predicting outcome or defining duration of therapy using this parameter. And also what an important question is the emergence of resistance And very important, mainly in our Latin American countries, is the price of the medication. Thank you.
So I'll start with a question. So we've heard about the challenges in some cases of even being able to use rituximab for CLL in certain countries. For example, maybe it's limited to just the FCR regimen. So what do you think the future holds for newer anti-CD20 antibodies in CLL? And I guess we have one example of that to look at right now, and that's obinutuzumab. So what do these drugs need to show to say that we should use them? Because they're certainly not going to be inexpensive.
Yeah. In Brazil, we have two scenarios because we have two kinds of treatment. The patients who are in private clinics, they can get everything. The patients in the public services, for example, we don't have even rituximab for these patients, for none of them. And then this is very difficult for us. I am participating in the obinutuzumab trial that compared obinutuzumab plus FCR, obinutuzumab plus benda, obinutuzumab mais chlorambucil. We didn't have benda. This is another point in our country because it was not approved by our regulatory service. I like very much benda, obinutuzumab. The responses that I saw, they were very good. But we have still to wait for further results.
Well, one of the questions that I have is that, when we look at both OFA and oblituzumab, they kind of push the dose. Rituximab's original dose was not an MTD, it was a dose that they could actually manufacture enough drug. So all of a sudden we started pushing the dose of CD20 and we see, a measurable but a small incremental benefit when we see these. My question is actually the other end of the spectrum. Can we go down? And in some of these, can you use a lot less of the antibody? Which then when you start talking about sub-Q administration, because again, rituximab is an issue because of how much you're giving. If you give a lot less, you can use it in smaller single syringes. Have we, and should we explore the lower effective dose of it? Again, in antihypertensives, we do that all the time. We give the minimally effective dose versus the maximally tolerated dose. So what about looking at that with CD20 antibodies now, and maybe the newer antibodies, you can get around and be just as good as rituximab with a lot less. I don't know. What's people's thoughts on, looking at the minimally effective dose that you may be getting benefit for these agents versus a maximally tolerated dose?
I think there are many issues we don't really know. I think you may think the other way around. I mean, the Germans have shown that elderly males do clear rituximab faster than females, and they advise that, or they suggest that elderly males should receive not lower dose of rituximab, but higher dose. But I don't know if really increasing the dose will be a good way to go.
Yeah, and well, part of the question is, well, will you get, effectiveness versus, again, because there's a, cost benefit ratio. So, can you get to points where okay, well, I'm not as good. I used to always give, again, being a solid tumor guy, I said, people give carboplatin all the time. Not that it's more effective or even as effective as cis, it's just so much less toxic that people say, okay, I'll take away a little bit of effectiveness for the tolerability. The question again in this is they say, well, yeah, I may lose a little bit, but I get a lot of what I get. Can I get 80% by giving, a third the amount of drug? I don't know. And that's just a question is, should we explore it?
John, I mean, if you look at the history of these monoclonal antibodies, there's been no dose-finding study. I mean, Roche never did it. The 375 came from multiples of 125. It worked fine on the shelf, that was fine. Nothing more than that. Now you're comparing, as you mentioned, rituximab to the other antibodies given at much higher doses, so it's not a real fair comparison. I mean, it's been shown that you can give higher dose of rituximab and it is more effective, so that is a bias anyhow. Clearly, I think, I mean, the point you raise is very important, because we heard from Valeria that, I mean, outside the private sector in Brazil, patients can't get rituximab. So I think, I mean, it would be better even to give a little bit rather than nothing. I mean, it's not very scientific, this, but I mean, in other areas, there have been data with very lower doses which are effective. So, I mean, it's something that hasn't been explored, and I think it would be worth exploring, because rather than giving nothing, I mean, it could well happen if this could be tested. On a different point, Valeria, why did Brazil, or the government, whoever, did they not approve bendamustine? I mean, what was the reason for that?
We have to ask them, because we can't understand.
But this is very strange. I mean, it's not a matter of costs, obviously. So what would the reasoning be behind it? You don't know.
No, it's a problem.
There is
suspect-
no explanation.
It's a
for
problem.
that.
Yes, the same. Now in Ecuador they are approving, but it was the problem with the cost of bendamustine, because bendamustine is very, very.
It's more expensive than
expensive.
chlorambucil, of course.
Yes, it's very expensive.